out there. Our next speaker is Dr. Mark Bartlett, and I've known Mark for the last 7 years that I've been involved with Pharmanex. He has degrees in biochemistry and organic chemistry, and right then and there, I was a chemistry major, and I know what that means, and wow. It was hard enough doing chemistry, but then he got a PhD in immunology and cell biology, so I really think there's something wrong with Mark. But he has another quality that is enormous, which is the ability to communicate. So you have intelligence and ability to communicate. And before joining Pharmanex, he was a visiting scientist at the National Institute of Health in Washington, D.C., where, get this, he studied the interaction of T cells with blood vessel wall and the role of various adhesion molecules that are used by these cells to communicate. What does that really mean? That means that Mark knows how things work. And things work with communication on the cellular level, hence nutrition, genetics, all those things play a role, and gave him the background that was perfect for helping us. And now, Mark is the Vice President of Global Research and Development for Pharmanex. I couldn't think of anyone better to speak this morning than Dr. Mark Bartlett.
Thanks very much. That was a nice introduction. Well, it's good to be here, and I've been here a couple of times before, and there's always a few new faces. That's great. So I'll be sort of talking more to the new faces, right? Even though I love my friendly old faces, young old faces. So I realize that you're busy people, especially the physicians amongst you. Some of you may still wonder why you're here. And what am I going to talk about today? I'm not going to talk a whole bunch. I'll tell you what I'm not going to talk about a lot of. I'm not going to sort of introduce all of our products and go through a product line or anything like that. I'm going to talk really more about our strategy as a company overall, and then to support that, some of the science that hopefully there are some touch points there and some familiarity, right? And if you haven't sort of gathered the message already, Nu Skin, when we sort of went through this period of self-discovery of who we really are and what we're really good at, realized that we're an anti-aging company. Now, what does that mean to you? What does anti-aging mean to you? Anyone want to volunteer that? Yes?
Slowing down the aging process.
Slowing down the aging process? That's good. A lot of people think, well, that's kind of a crazy notion. There are gerontologists all over the world who have been studying aging. It's really only become popular as the baby boomers have aged. Actually, there weren't, 30 years ago, not very many labs in the world at all studying the aging process. Isn't that interesting? And there is still sort of the pervasive idea that you really can't slow the aging process. Most gerontologists really are just into sort of observational biology. They will tell you the things that do change as you age, whether it's neurological or musculoskeletal, the things that change as you age, what you expect, and we can explain it all, right? So this notion of actually being able to slow the aging process is fairly radical. Anyone believe that it's even possible? I hope that today, really, I want to sort of share with you what our strategy is at Nu Skin and how we approach this concept of anti-aging. There's maybe another concept that I should mention. When we think slowing the aging process, we often think then, how long are we going to live? Are we going to live to 110, 120? That is an interesting concept, and maybe that's possible. But what we're more interested really in is dying young, as late in life as possible. You know what I mean? That's sort of the popular way of saying it. The doctors would say compression of morbidity. You know what I mean by that, right? So morbidity meaning sort of that period of sickness before you die, those years of incapacity, whether you're in a wheelchair, you're on oxygen, or you're just not living life to the fullest. So that's morbidity. So you want to compress that, right? So even if you only live to 75, if you're healthy and happy and youthful all the way to the end, then that's sort of where we need to be. So it's more a focus on compression of morbidity than really longevity per se. So let's talk about our approach then. I'll talk a little bit about nutrition and free radical biology. the role of phytonutrients in gene expression. That'll sort of come toward the end. And, wow, am I going to talk about mitochondrial aging? Yeah, I guess I am. Let's talk about that. Okay, so here's the first sort of, when we look at a Venn diagram of aging and maybe affecting the aging process, the first thing that comes up is nutrition. Really, that's the lowest hanging fruit. What? I see you smiling. Do you think that's an obvious place to go? Yeah, it's really, really some low-hanging fruit there. So look at percent of adults that are not meeting the recommended daily allowance, and we're talking here of micronutrients. Okay, so mostly talking about micronutrients. I think our bodies are fairly, fairly adaptable, depending upon where you go in the world. What's the major macronutrient? Is it fat or protein or carbohydrate? We can sort of get by there. There are some issues there when it comes to body shape and weight management that I'm not going to cover today. But when we focus on the micronutrients, look at how many people are not meeting the minimum requirements. Doesn't that astound you? Look at zinc, for example. 80% of females not meeting the minimum level. What does zinc do? I don't have, sorry? Immunity amongst other things. Zinc is actually a critical component of DNA, because it actually is, sort of is a glue in DNA structure and also in protein structure. So there are over 500 enzymes that require zinc as a cofactor just to hold them together. Now, one example is DNA polymerase. So immunity. When the latest flu bug comes along or cold comes along, your body has enough T cells, really just, it has sort of this army of T cells that are all quite different. And most of them are never actually used, right? But when an antigen comes along and it has the right sort of antigen for that particular T cell, what happens? That T cell alone can't fight that particular antigen, but it actually sends a signal to the cell. There's T helper cells, there's B cells, right? And they work together and basically then make a clonal army of that one T cell so that you can fight the disease and then get immunological memory. So in order for that to happen, you have to have copies. You have to make a clonal army. And when that happens, you need this enzyme called DNA polymerase that helps make multiple copies of it. If you're short, if you're marginally deficient in zinc, then how can your immune system build that clonal army front line to fight the latest bug that comes along? So, that's one example. And look at how many micronutrients you're deficient in. This is the average American. And they do this study about every 10 years with the USDA CSF studies. Right now at BYU, there are about, as I drive past this particular part of BYU, they've got about 6 semi-trailers parked there. It's kind of like a Walmart parking lot. And this is all the USDA CSF studies that are going on sort of around the world all the time, or around the US. So they're doing this about every 10 years, and what changes? Pretty much nothing. Okay, so it's an indication that there are marginal deficiencies there. Well, how important is that? If you really want to get a handle on it, Bruce Ames is doing the most comprehensive and systematic studies in this. Bruce Ames you've heard of, right? Yeah. He's famous for the Ames test, so virtually any new thing that's introduced into our diet actually goes through this Ames test that he invented about 50 years ago that looks at micromutagenicity. So we want to make sure that nothing that goes into your diet actually sort of damages the genome. But lately he's been studying more micronutrient triage. And the basic concept is that if you're marginally deficient in a nutrient, then your body will have to decide the most important place for that nutrient to go. It has to make a decision, and it's like a triage. So, for example, there are things that are required by that nutrient to keep you alive today. Let's look at calcium, for example. What do most people believe that calcium is important for? You don't get enough calcium, what's going to affect? The bones. Right, that's the first thing we think of, but actually calcium is important in nerve transmission, right? If you don't have, calcium flux and all of these things related to calcium and magnesium and these electrolytes actually affect how we think and our nerve transmission. I can't move my arms or even think without calcium. And so if you're marginally deficient in calcium and there's a triage going on, what does the body decide to do? Well, if you're going to be able to survive, you need to be able to think and move. And so the calcium that you need will be focused in on those systems. And it will be at the expense of bone health, which you will notice when? Later in life, right? And so this is a really good example of how this micronutrient triage affects aging. That if you're marginally deficient in these nutrients, the body will make a decision to keep you alive today, but it will be at the expense of aging well. So, again, he's done systematic studies. So calcium, magnesium, good for cognitive function, think and survive, fight and flight, later on bone density. So I think most of us, even if we made the conscious decision, would say, yeah, I'd rather have osteoporosis later in life than sort of get run over by a bus today because suddenly my nerves sort of froze up, right? And then if you look at it sort of more on a enzyme cofactor level, you can even identify all of the different genes and gene products that are necessary for short-term versus long-term survival. And this is the kind of work that Bruce Ames and his workers are doing, and others as well. But when the availability of a micronutrient is inadequate, the body ensures that micronutrient-dependent function required for short-term survival are protected at the expense of functions whose lack has only longer-term consequences. It all makes sense, but if we want to age well, if you want to help your patients age well, then this concept of getting enough micronutrients and not being marginally deficient, as most Americans are, should perhaps be one of your number one goals, right? If you really want to be a force for good, this is great knowledge. And I encourage you to maybe look up some of this work. Go to Medline, Google micronutrient triage, Bruce Ames, read a couple of those papers. They're very enlightening. All right, what's next? If we were to put nutrition science first, what might come next if you're looking at sort of the aging strategy? Yes? Genetics, I'll put that third in the bucket. I think even before genetics, I would put antioxidant science. Would most of you agree with that?
Yeah.
There's an overlap here too, because a lot of nutrients are also antioxidants. So the Venn diagram is I don't know how much it overlaps, but there's a fair bit of overlap there, right? This is where Lester Packard came in, the father of antioxidants. He's one of my heroes. And, we get to interact with him a lot because he is on our scientific advisory board. And I asked him one day, I sat down and said, what does it feel like to be the father of antioxidants? He says, well, by now I'm the grandfather of antioxidants. So, and it doesn't seem all that radical to us. No pun on the word radical here. But really it was a radical departure from sort of mainstream nutrition. When he started to posit this idea of free radical biology, in the 70s or early 80s, it was just starting to catch some traction. And what's unique about this approach is he's talking about subatomic particles interacting with biological molecules. So this is really nuclear sort of science interacting with biology. Are you hearing okay? Oh, I'm going to go closer. Okay. Barry White coming up. So, this is a whole departure, and a lot of people at first thought this is kind of quackery, this idea of, subatomic particles interacting, but really that's what's going on. So the same kind of damage that you see with free radicals on cell systems is pretty much the same exact mechanism that you'd see if someone was exposed to radioactivity at a Chernobyl meltdown or something like that. So, antioxidant science. Where do these free radicals come from? Pollution, smoking, of course, so don't hang around the chimney stacks and certainly don't make yourself a chimney stack, right? Stress can cause free radicals. Sunlight, did you know that? Just ordinary sunlight. Not necessarily just even the UVA and the UVB that we know is damaging and aging and burning, but light itself, interacting on the surface of your skin, actually generates these reactive oxygen species that then result in damage to the immune system. We have dendritic cells and all sorts of cells at the surface of the skin that are immune related, and it actually suppresses immunity and damages the skin. How does the body fight back? quite nicely, right, with the melanocytes producing this pigment called melanin. But, even just sunlight itself can cause free radical damage. So we need to prevent that. Here's one that people don't often think about, is internally. Anyone know what that sausage-like structure is? What that represents? That's the mitochondrion, right? The mitochondrion is a marvelous thing. Every cell has a few hundred of these, and their job is really to combine fuel with oxygen to create ATP, which is the chemical storage form of energy. And as the mitochondria age, there's a little bit more metabolic exhaust. That's basically free radical, because part of this process is using oxygen as a final electron acceptor. So we have subatomic particles there, free radicals, that are being generated. If it becomes less efficient, then that sort of gets worse and worse. So just metabolism in the body creates free radicals. But don't worry, the body does produce internal or endogenous antioxidants. We'll talk about that in a sec. So here's the cell membrane again. And this cell membrane, you've seen the picture, right? As Dr. Moore showed it up here. The lipid bilayer and all of the cell communication molecules in the surface. These are some of the touch receptors and things that I was really interested in when it comes to cell communication. The way that cells communicate is they touch each other and they have a certain level of stickiness, right? They have these selectively permeable pores where small molecules can pass back and forth between the cells, so there's sort of a chemical communication, a little bit like ants touching their feelers, right? And they have those scents. And also the cell fluidity does affect the communication. So we don't want to damage that whole system, and free radicals tend to do that. They tend to damage the lipid bilayer, and that causes a stiffening of the cell wall. So we need to protect it, and the way that we do that is with antioxidants. Here's something that may be very familiar to you. Anyone seen this sort of theory of oxidized LDL in the blood vessel wall before? And this is all about atherogenesis. A few people nodding. Let me give you a story. It was when I first started looking at antioxidant free radical biology in the 80s. And I was doing some work with LDL, and we were measuring LDL oxidation. And my dad was turning about 50 at the time, I suppose, and he came to me and he said, "Mark, I've been healthy all my life," and he's a 6'2", strapping Aussie male with big muscles. I was the runt of the litter, by the way. He has 10 children, and all my sisters are the same height as me, and all the boys are like 6'3" and 6'4" and big muscles, and I think I had a knot in my cord or something. But he said, so for the first time I've gone to the doctor and he said I'm not completely healthy. I'm like, "What are you talking about, Dad? What is it? What is it?" He said, "Well, my cholesterol levels are high." And so the doc wants to put me on this statin or something. And I said, "Well, let's look at that a little more closely. Dad, did you know that if you have good antioxidant levels, then that LDL particle is not going to be oxidized, and therefore it will not even be recognized by the blood vessel wall?" And he said, "What the hell are you talking about, son? Speak English." Right? Well, that's the story, right? That you have these LDL particles. When we're looking at cholesterol, basically we're looking at HDL or LDL cholesterol molecules, and the science does support this concept that if that LDL particle is not oxidized, then it does not become atherogenic. So you can have, theoretically, cholesterol levels that are through the roof, but if they're not oxidized, then you don't get cardiovascular disease. Perhaps Dr. Michael Jason might agree or disagree with that, but it's basically the concept, isn't it?
1,000%.
Yeah, we have a cardiologist here, so he's backing up my words. So this is the concept. Yes, maybe it's important to lower cholesterol to a good level, but the other half of the story is purely nutritional. Make sure that you get enough antioxidants, and that's why there are so many studies that show that people who eat high levels of fruits and vegetables, Mediterranean diet, whatever it may be, have low levels of oxidized LDL and have lower incidence of heart disease. You may think that the big story with fruits and vegetables is cancer. The big story is cardiovascular disease. Cancer is there as well, but number one on the list is cardiovascular. So antioxidants do become important. So we've already talked about the carotenoids, and wow, I don't know what's going on with my... You can't really see that, can you? I was going to show you some it's almost pornographic how beautiful these lycopene, lutein, beta-carotene molecules are, but I can't even see them here. These are, if you've looked at the structure, it looks like a zigzag on the page, right? And every zig, not a zag, has got this 2 lines instead of 1 line. Any chemists in the room know what that means? When every second one is a double line? It's to do with, they're all covalent bonds, but this is a special, I think it's, it's about orbitals and it's about conjugation. Basically what it means is that you have this cloud of electrons that can actually locate across the entire length of the molecule. So yeah, it's covalent bond, but then there's some electron sort of uncertainty going on there. Basically what that means though is that you have a cloud of electrons along these molecules that you can't see, and that frequency of the electrons buzzing back and forth is actually what gives them their color. So when you see the red, the orange, and the yellow, it's the chemical structure of that carotenoid that's doing that. And it's the same thing that's giving it color is also giving it the ability to absorb hits from free radicals without damaging them until they get at least 20 hits, and then they become damaged, right? So that's what makes them such a good first line of defense for protecting the cell membrane, which includes the nuclear cell membrane here, which I haven't shown being protected, but those carotenoids are lipid soluble. And we even know it's not just sort of artistic license that I've used here. I created this slide about 15 years ago, and it still seems to be on the circuit. It's kind of funny, isn't it? But this is not just artistic license. You see how the lycopene, the red one here, is vertical along the membrane? It's because of the length and its lack of any polarity at all. Then you've got some that are oblique across the membrane and some that float, like lutein. The yellow one has polar ends that swivel, so it actually floats like a raft in the cell membrane. So these all provide amazing defense against free radicals in the cell membrane. And I said I wouldn't talk too much about product, but we've heard about the scanner. We'll talk a little bit about that in a sec, and how it can detect carotenoids in the skin. And we've talked about the importance of antioxidants and how you need to maybe increase your score. How do we do that? Well, we could do that, I guess, by giving you a supplement that just has a massive hit of beta-carotene. It could be synthetic, it could be natural, wouldn't matter. It would drive your score up. So, what's wrong with that concept? Why wouldn't we choose to cheat, maybe, and give you a bottle of something, beta-carotene, 50,000 IU, guaranteed to raise your score? Is that all we need to do? Is that the goal?
No.
Thoughts on that?
Imbalance.
Yeah, it's not a balanced approach, right? When you eat a diet...
Raise the score and kill the patient.
Yeah, maybe. In fact, there are some studies, right? You may remember a couple of studies that showed that amongst smokers, high levels of beta-carotene proved to be more carcinogenic instead of protective. Remember that study?
And explain why that study can sometimes be out of context.
Yeah, it can be out of context because you talk about one level. I think they used 60,000 IU of beta-carotene in that study. There was a synthetic beta-carotene. And beta-carotene is kind of unique among the carotenoids because it actually, its metabolites are bioactive. And in the case of smoker's lung, where nicotine sort of revs up metabolism, and you have this bioactive retinoid that results in high beta-carotene levels, then it adds up to abnormalities. Problem number one is not the fact that the guy was eating a lot of carrots, but he was smoking the carrots, right? So that's, it's kind of funny how it turned out that the studies made it look like beta-carotene is the devil, when actually smoking is the devil. Stop smoking, right? But when you have, but the context is good. When you have normal levels, let's say a diet rich in fruits and vegetables, you're getting not just a mega dose of beta-carotene, right? You're also getting some of the cis and trans isomers like and alpha-carotene. And you're getting a lot of these other carotenoids, and you're getting vitamin C and vitamin E. So you're not sort of in this state where it's abnormal to have huge levels of one carotenoid.
This concept that Mark is talking about is really the differentiating factor on how physicians are trained when it comes to studies. In studies, we're always told, what compound is it that makes a difference in the study? Is it the vitamin C? Well, the vitamin C doesn't work. Okay, is it the vitamin E? Well, the vitamin E doesn't work. Therefore, antioxidants don't work. But it's actually a combination. Life is one symbiosis. It's a combination of various things acting together, and that is why nutrition works.
Right. Right. So we want to try to copy nature here. And so you need to know that our approach has always been, even before the scanner came along, has been let's copy nature and let's look at, not just one level of an antioxidant that's super high, but what would you get in a diet rich in fruits and vegetables? What are the levels of lycopene, lutein, beta-carotene? What are the levels of the flavonoids? And the flavonoids are some of these other antioxidant molecules that you find in, especially in the skins of the fruits and vegetables. Some of them have color. So if you look at a beet, for example, this beautiful purple color, not much in the way of carotenoids there, but these are flavonoids. And the flavonoids will have names. There's families of them, the anthocyanidins and the proanthocyanidins, and there's a whole bunch of chemistry going on here in the foods that you eat, the phytochemicals. So we want to have a balanced level for nutritional support in all these different areas. Did we see this already? I can't remember if Stephen showed you this. So we did a study at the University of Texas with GLL and some experts in antioxidants. And we looked at, first of all, if you give someone LifePak for, I think it was a 12-week study, are the levels of these antioxidants and other things increasing in the blood? And it's important to know that they are. That's great. But are they actually bioactive? And here's a study on LDL protection. Remember I mentioned that earlier? So lipid-soluble antioxidants should theoretically be able to protect LDL from oxidation, which is exactly what you want. In this experiment, you actually take the blood, and I've done thousands of these experiments. I've hung around blood banks till I'm blue in the face asking patients for an extra 40 mils of blood. And I take it up to the lab, and you have to ultra-centrifuge it for about 12 hours under refrigeration. We're talking 60,000 RPM sort of centrifugation. You get everything out except the LDL, which sort of floats to the top. And then when you've got this pure LDL, you then can challenge it with copper sulfate or some really potent oxidant, and then measure when it's being oxidized over time. So that's what we did in this experiment here. But instead we had a control, or people taking LifePak. Understand? So you have someone taking LifePak, someone taking for several weeks, and someone taking no supplement at all. Isolate the LDL and see how it stands up to oxidation under these laboratory conditions. So it's an ex vivo experiment. Human study, take out the blood, get the LDL, see how rigorously it can defend itself against oxidation. And usually, you don't see much. I mean, it's not very easy to help sort of stop this oxidation. But you can see here that those people that are on LifePak, this is basically time, and they were able to protect themselves much longer, about twice as long before it was oxidized. So it's a really nice study that showed that there is an effect. Okay, why do I show a picture of this is a hearse with some flowers. And really, when it comes to showing studies like this and this, if you're a doctor, how often do you show the papers to your patients? And how often do they go out of your office going, woohoo, I understand it now, this is awesome, I'm going to take antioxidants. I saw that bar graph and it really meant something to me. Is that the reaction you get? No, it really isn't, right? There's these young ones sort of getting very sleepy in the back here, and I don't blame them. And that's what your patients are like when you try to show them graphs like this, right? So you have to do something that's meaningful for them. You have to give them an emotional response. Because most people will not change their lifestyle until the hearse is backing up to the front door and they're smelling the flowers, right? And it sounds funny, but how many times have you seen this, right? My sister smoked like a chimney until she had a perforated duodenal ulcer and nearly died. And that caused her to stop smoking for about 5 years, and then she was back on it, right? I mean, habit is a powerful thing, and you've got to change those habits, and you've got to get people to follow a regimen. And it's hard enough to keep people on a pill like a HMG-CoA reductase inhibitor, isn't it? Let alone sort of changing their diet and their lifestyle and thinking about antioxidants. So how do we approach that? We want to be a force for good, and this is where the scanning comes in. So I won't go into great detail, but here's another thing that you can Medline search. how are carotenoids involved in the aging process? And now it's actually amazing. You Medline search carotenoids, you will find thousands of studies. And I've just sort of shown a few here, where from newborn, from neonates right through to old age, carotenoids are attached to age-related biomarkers and factors. It's really quite incredible how important carotenoids have been. It doesn't really seem all that logical. These are antioxidant molecules that somehow are tied in with all of these things. Low serum concentrations of carotenoids and vitamin E are associated with high adiposity in Mexican American children. We know that it's a biomarker of fruit and vegetable intake, so maybe there's a clue there. But even predictors of fetality in neonates requiring mechanical ventilation. There's a correlation between the carotenoid levels in their blood and how serious some of their ailments may be right after birth. And then we have lots of studies in aging individuals. For example, they'll get a whole bunch of octogenarians and divide them into quartiles of their carotenoid levels, follow them over the next 10 years and see who dies first. And the ones with the highest level of carotenoids are there until they're centenarians. It's really quite interesting. So look up some of those studies. It's fascinating. So we have the S3 now. There's almost no excuse not to be scanned. What's it all about? Well, I won't go into the science. I don't know. How am I doing on time here? Anyone got the time? 10 what?
10:08.
Sorry?
10:08.
10:08, thank you. It means I've got a few minutes left. It's Raman spectroscopy. C.V. Raman won the Nobel Prize for this technology back in 1930. Did I ever tell you that some of his nephews or great-nephews visited the lab one time? We had these two Indian guys turned up at the lab, and they said, my last name is Raman. And Raman, that sounds familiar. There was this guy named Kembat Raman, and one of the guys says, my name is Kembat Raman. And I said, he was my great uncle. And I said, well, he won a Nobel Prize, didn't he? And the guy said, no, he won 2 Nobel Prizes. So it's kind of interesting, isn't it? They were very proud of their great uncle, and he did amazing things. The guy had a master's degree by the time he was 15 years old.
Wow.
So brilliant man. And Raman spectroscopy was something that I remember learning in organic chemistry, but there was just 1 page on it. And do you know why there was only 1 page on it? Because nobody was really doing it. It was recognized as a brilliant form of spectroscopy, but even when I learned this in the 70s, there were no lasers that were readily available, and there was no personal computer that was able to do the algorithms that were necessary to separate the background from the peak. So it was these brilliant scientists at the University of Utah Biooptics Department. Basically, they're a bunch of geek physicists that were able to figure this out. And again, it was really just a greenhouse technology. The laser was 4 feet long. It was an argon laser. And before the laser could work, it had to be super cooled with liquid nitrogen down to minus 70 degrees. And so once they poured the bucket of liquid nitrogen over the laser and got everything set up and calibrated, then you could put your hand in front of this 4 foot long thing, and you could almost smell the bacon as you put your hand in front of it. And so it cost our company, as we became interested in this, $70 million to shrink the technology down to what we have today. So we've made it accessible to people. And why? Why have we made it accessible to people? It goes back to that picture that I showed of the hearse and the flowers. It's because you need an emotional response before you'll change your behavior. And we don't want that emotional response to be a result of a major health event. And you can do that. It's really quite amazing. When someone gets scanned and they get a number, if they have a context for what that number means, then it creates an emotional response. Sometimes it's anger, sometimes it's denial. You sort of see all the steps, right? Sometimes it's just pure competition. When my daughters were first scanned as teenagers, I have 4 daughters, and some ran and some did track and some did cross country, and when they got scanned and it was kind of low, suddenly there was this competition between the sisters. How do we get our score up? Suddenly they're asking me questions about whether or not fruits and vegetables or carrots are better when they're steamed or raw. What's going to make my score higher? Anyone know the answer to that? Steamed carrot or raw carrot? I'm hearing raw, I'm hearing steamed. The rawtarians are going to be really disappointed in this answer. Steamed is actually better. Sometimes you need a little help before you can break the cell open and release the carotenoids for higher bioavailability. So, cooked tomato with a little bit of oil actually not only helps break open the cell structure and give you more lycopene, but the oil helps with the bioavailability. So don't throw away a healthy salad oil for one of those fat-free ones, because actually a little bit of oil helps with the bioavailability of those carotenoids that are in the salad, right? So it becomes an emotional response, and you can see, this is the result of over 7 million scans around the world. Look at the data that we've been able to generate, showing that smokers score poorly. It damages the first line of defense, right? These antioxidant molecules. The obese also score sort of close to smokers. If you're interested in understanding more about that, we do know that diets that are high in sort of unhealthy lipids can actually cause an increase over the short term, couple of hours, of these inflammatory molecules. So look up Paresh Dandona, D-A-N-D-O-N-A. He's a scientist from New York. He's the guy that did that take McDonald's meal and then follow the free radicals and the inflammatory molecules like NF-kappa B over a period of hours after the meal, and you see them rise dramatically. So chronic overeating of unhealthy foods actually causes free radical damage in the body. And that may be why we're seeing, it's not just the dilution effect of the lipid in the adipose tissue, but more is going on there. Then you've got, so these are sort of the average Americans around here, 2 servings of fruits and vegetables or less, and then it goes up from there. Now, some people do ask me, what's an unhealthy, can you overdo this? Can I have a score that's too high? Let me just maybe throw in one anecdotal story to help you understand the context of this. I have a daughter who, this is kind of crazy, but when we were really doing the pioneering work in the scanner, of course my daughters got scanned a lot and they became very interested in their scores, and they actually, 3 of the 4 became vegetarians. 1 is a vegan, but that doesn't mean that they just eat sort of white bread and cheese. They actually sort of really became healthy, and they started cooking their own stuff. One of my daughters even sort of ferments her own kombucha. Anyone know what that is? Yeah. So, just crazy stuff. My daughter, Stephanie, who works in the lab, doesn't take LifePak because she's vegan, so she doesn't want to take the gelatin. But her score is typically over 100-something thousand points. So do you think she's unhealthy? Do you think she's overdoing it? She looks young. Since she sort of started on this sort of radical vegan diet, she actually looks great. Her skin has a nice tone. She's just really looking healthy. So I would say that you're not overdoing it unless someone comes up with a supplement with ridiculous amounts of beta-carotene just to pump up your score artificially. You've got to keep that balance there, right? And that's what we've always striven for. So far then I've talked about nutrition science, I've talked a lot about antioxidant science, perhaps taking up more time than I should have, because I want to talk a little bit about the next circle in the Venn diagram, which someone who already brought up. It was genetics, right? What role does it play, I guess is the question. Is it outside the circle or is it inside the circle? What role does genetics play with aging? What do you think? Some people age well, right? Some people age not so well. We look at our parents. The Laotians have a saying in their country, "Our mea dong bun man," which means, if you want to choose a good wife, you need to look at the mum. Isn't that interesting? And it's kind of true, right? But there's a problem with that theory. What is that? You can't change your DNA, right? Nu Skin is not interested in doing sort of weird genetic splicing experiments, right?
Gene expression.
Right. So the DNA is fixed for life. Let's sort of look at genetics 101 here. The DNA, here's the molecule of DNA. You can see here the chromatin structure, that's the chromosome, right? And you unravel that and you get DNA, and at points across the DNA you get the gene, right? So you've got your A, T, G, and C, these bases. Does it change? No. You don't want it to change, right? I know that there's There's an issue with genomic stability. It's being attacked all the time. And some of the work of Bruce Ames and others looking at marginal nutrient deficiencies shows that the importance of making sure you get enough of these micronutrients is to do with genomic stability. Did you know that? You knew that part, right? DNA is always being broken down by free radical attack. So you have to resist the breaking down of the DNA, you have to resist these nicks that are constantly being introduced, and you have to repair them. So we have a lot of these basic micronutrients, B vitamins, are actually cofactors for enzymes that are DNA repair enzymes. So again, if you want to keep your genes stable, here's one by Fenwick, an Australian scientist who studies the micronutrient relationship with genomic stability. So Fenwick, F-E-N-E-C-H. Michael, I think is his name. CSIRO, that's the Australian version of, I don't remember the name of it in the US, but anyway, look up that paper, really interesting. So we want to keep genomic stability. So what's the relationship then? We don't want to change the DNA, and as you already mentioned, so it's about gene expression. That's something that we can influence. So looking then as far upstream as possible, we've already looked at micronutrients, we've looked at antioxidants. There is a role for genetics, and it's in the area of gene expression. So plant chemicals, we've talked about those today. You know, the carotenoids, the flavonoids, all those things. It's really fascinating stuff. It's not just the macronutrients here, and it's not just the micronutrient vitamins in the plants, but it's these other molecules that are bioactive that we're really interested in. And for the most part, we thought that they were just antioxidants. But it seems that they're much more than antioxidants. And as we study them more closely, we realize that they have effects on gene expression that we didn't even realize. So here's a carrot, not to scale. It's there to demonstrate that carrots contain beta-carotene, right? And so here's the molecule, sort of the cell surface again. You can see the lipid bilayer, you can see one of the gap junction proteins, and you can see here free radicals that are roaming unfettered in the cell, trying to damage the cell, right? So the carotenoids are helping to prevent that damage, but something else happens. When the carotenoid is damaged, then it sends a message to the nucleus which actually affects gene expression and upregulates endogenous antioxidants in the cell. So you see what happened there? There's a message to the genes somehow. We don't have to go into all the mechanisms that are going on there. It's probably actually multi-step. But ultimately, a transcription factor arises. A transcription factor is something that is able to turn the dimmer switch on the gene up or down. And in this case, that transcription factor, which resulted from the presence of those oxidized carotenoids, turned up the dimmer switch on the gene that produces an antioxidant protein. So what's brilliant about that is that the beta-carotene molecule has not only prevented sort of further damage from antioxidants, from free radicals I mean, but it has also become sort of a litmus paper for free radical activity in the cell that has then asked the cell to adapt in a genetic way. That's gene expression. So it's kind of cool, isn't it? Let's look at another example here. So here is this molecule. I haven't talked too much about it. Remember, it's the gap junction. And if there's another cell here, these often line up. And so small molecules travel back and forth between the cells, and this is a part of cellular communication that enables a cell to act sort of as part of this community of cells in the body. And so we want that. And in actual fact, in a cancer type cell, you have less of these communication molecules. What does that tell you about cancer cells? They really don't want to be part of the community. They actually then start to have uncontrolled growth, and they genetically sort of become less differentiated, right? They sort of become more stem cell-like. So all sorts of weird things happen when they don't communicate properly. So here's a tomato, again, not to scale, but what's the red molecule that tomato is famous for? Lycopene, red carotenoid. So it seems that in the presence of more of these lycopene molecules, another message is sent to the nucleus to say, make sure we have enough of these communication proteins on the cell surface. So there's a correlation between those carotenoids and the health of the cell in terms of its ability to live in the community. So we're seeing here another side of these plant molecules. They actually do more than just act as antioxidants. They can send signals to upregulate genetic expression of endogenous antioxidants, and they can influence cell-cell communication. So we see that there's gene expression stuff going on here. So how does that apply to new skin and aging? Well, we're really interested in aging. Both of those things that I talked about, cell communication and antioxidant capacity, can affect aging, but is there more to the story? And we were very curious about that. For years we'd looked at gene expression, genetics, we looked at SNPs, of course. There are imperfections in the human genome, we know that. There are about 1 million that have been identified. Every individual here, I know that you think you're perfect, but we have about 30,000 what you'd call SNPs, or single nucleotide polymorphisms, imperfections in your DNA. Maybe Adam had 0, Eve had 0, but over time, with all the nicks and scrapes and things, 30,000 in the average person. Most of them don't actually affect you. Occasionally, that single nucleotide polymorphism change can then lead to a problem in the enzyme, or that then can give you an inherited disease. So that's what that 23andMe is all about. It's all about SNPs. Again, we're not about that. We'll accept that that's the case, but we're more interested in aging and gene expression. So some of the most relevant research in this area, we thought, and brilliant sort of a new area, was these guys right here. Here's Richard Weinrich, a gerontologist, Thomas Froehler, who's a geneticist, and then Jamie Barger, who's worked with them, also a geneticist. And they sort of came up with this new paradigm. There is a technology that's already available called the gene chip, right? The gene chip measures the transcriptome. So when you go back to the DNA, when the dimmer molecule is turned up on a gene, what happens? It makes a lot of copies of that DNA. It's called messenger RNA. So lots and lots of copies are made. And that messenger RNA is transcribed from the DNA. It's a copy. It's very much like the DNA. It's basically a mirror image. It uses a different, one of the base pairs is different. But then lots of copies of that messenger RNA then are translated into proteins, gene products, right? So that's what gene expression is all about. The transcriptome is how much RNA there is, and that's measured by this gene chip. And it's an amazing technology, because in a less than an inch square of this silicon chip is implanted about 40,000 probes for all of the known genes that we have. There are about 25,000 genes, and there are about 40,000 probes there. So you can get a purified sample from human tissue, extract the RNA, put it on the gene chip, and it will light up. So where you've got a lot of messenger RNA for a particular gene, it'll be bright, and where there's not much, it'll be dim. So that way you can actually measure what's going on in your body, what's being expressed and what isn't. If you had your carrots this morning, is that gene for that endogenous antioxidant going up? It will be, right? So what they did then was basically they compared two biological states, young versus old. And they zeroed in on the gene expression pattern that change. Not all genes change as you age, but some are. And when you can zero in on that, now you have a way of measuring aging at the genetic level. And once you can do that, what's the next step? Sorry? Yeah, you want to reverse it. So how do you reverse it? What would you do if you were a scientist at Nu Skin and you had this technology? You can measure aging at the genetic level. Would you be curious about what's a good strategy? Maybe you'd look at stress versus not stress. We're really interested in plant chemicals, right? Phytochemicals. And so we tested a whole bunch of them. We wanted to know, are there some that can change? Well, the first thing that we did, we sort of narrowed down our search. There are 25,000 genes, and aging is sort of a big thing. So we narrowed it down to mitochondrial activity. We know that as you age, mitochondrial efficiency declines. So that's all to do with your energy, right? And this is one of the first things that you feel as you age. So we looked at this. Here's a picture of a cell here. You can see the green is stained with these mitochondria. So the cell is full of these mitochondria producing energy. As we age, the studies show that we reduce in effectiveness. Do some of you feel that? Any in the 61 to 84 range in the room? I know that I'm not 61 yet, I'm 53, but I've felt the decline, so I'm somewhere around here, right? You feel the energy levels going down. So we looked at gene expression of the mitochondria and zeroed in, with the help of LifeGen, of course, on which genes are changing that make and maintain the mitochondria. So we have mitochondria actually have their own genes, but most of the genes to do with building and maintaining the mitochondria are actually in the nucleus. There are about 1,000 genes that have been identified that basically build the mitochondria. So looking at those 1,000 genes and looking at old versus young, here's what we found. About 52 genes change in expression. So here, these are expressed at a low level in the young, at a high level, and then as we age, it flips, right? So those are the 52 genes that we identified that change dramatically in expression as you age. Now we wanted to test for ingredients, an ingredient panel. And that's where we're kind of experts, okay? Our LifeGen technology may be the geneticist, but Nu Skin has been dealing with anti-aging phytochemicals for 30 years. So we knew what concentration to look at and what some of the most promising things would be. And some of them came from traditional Chinese medicine. So here is an example, 5 of the 50 or so that we first tested. We tested ingredients in all individuals to see if that would reset gene expression back to a younger state after 3 months on the diet. So what do you see there? What pops out at you? So we want this. Everyone that tested started off like this, right? We wanted to go back to this. This one good? This one? This one? Yeah, indeed. Kind of dramatic, isn't it? That you can change gene expression. So in this case, the theory is that we are changing gene expression back to a level of youth in the case of the mitochondria. And the ingredient that we saw there in that panel was Cordyceps sinensis, kind of an unusual mushroom that grows only in the high plateaus of Nepal. And it made the yak very frisky. It gave them sort of a sexual vitality. But the horses that fed off the grass that had that mushroom sort of poking up through were also famous for their vitality, for their stamina. And so Cordyceps soon became famous amongst the Chinese. It was worth its weight in silver. And it was a sort of an anti-aging vitality type ingredient. So it's interesting to see in modern science how it sort of lit up those genes, isn't it? What was going on there. We're also interested in cellular renewal. This is not something that you necessarily feel so much directly related to energy, but cells have to be able to recognize components of the cells, proteins, enzymes, structural proteins that get ragged and old, right? That need to be replaced constantly. And so there are a whole bunch of recognized mechanisms for detecting sort of worn out components of the cell and then either repairing or replacing. So those components of the cell we also looked at in the same sort of platform. And here's the gene expression profile for the purification. This is cellular energy, which you already saw. Here's purification. So the same kind of platform was used to screen for ingredients that would be effective in that area. So I have about 5 minutes left. This translated to, when we looked at mitochondria, for example, we did a study that looked at mental cognition. I don't know if most of you realize it, but it's something that we found out as we started to study mitochondria, is that a lot of conditions when it comes to the aging brain are related to the effectiveness of mitochondria. So, scientists that work in the neurosciences that want to get a medical model of dementia, what they usually have is some specific toxin for the mitochondria. It's interesting. And so they have this toxin that's specific for mitochondria that when they use it, it triggers what looks exactly like dementia. So you start to realize how important energy is in the brain when it comes to cognitive function. So it's really the canary in the coal mine when it comes to aging. So the mitochondria are really important, and we thought, well, maybe the first place we should look in humans is the brain and cognitive function and see if this is making a difference. So the 3 ingredients that we found to be most effective with mitochondrial gene expression, we put in a formulation and then tested over a period of 12 weeks using some sort of cognitive tests. I actually was part of this study just as an outsider to see how it was going, and yeah, it was kind of challenging. You know, they'd flash up 5 cards and then they'd take them away, and then they'd flash up a card and say, was that the right card that was in those first 5, and is it in the right spot? You know, things like that. So there were various decision making and short-term memory, long-term memory type things. And look at what happened over 12 weeks. So here is the control group, and they improved. They improved quite a lot over that period of 12 weeks. That's to be expected, right? Because we should exercise our brains, and as we do, and as we practice, we get better at stuff. And so that's what happened. But look at the ones that were on this formulation. They improved faster, and at the end of 12 weeks were at least 20% better at those tasks. So that's an example of what we've done with gene expression science. We applied this to weight management science, looking at a bunch of things. I won't go through all of that. So let's summarize then briefly. If we're looking at aging, and if we're interested in aging, what are some of the things that really stand out when it comes to nutrition? And what can you apply to your patients? First of all, nutrition. It's extremely important, right? There's a micronutrient triage sort of concept that is now systematically being looked at, and it seems to be very rigorous. So we need to make sure that we're not marginally deficient in any of the nutrients. Not just for the sake of our immune systems or anything else, but so that our body can not have to make this triage decision. We basically filled the hospital with doctors and nurses that can take care of everything that comes along, right? Second is antioxidant science. Understanding that we need to protect the DNA, genomic stability, but also that we want to prevent the damage caused by free radicals. And to be able to measure that with a non-invasive device that changes the paradigm from boring science to emotional response so that you can change your life is a good thing, right? And that overlaps then a lot with the genetic science, because what we're looking at is gene expression science. And that ties in very closely with your fruit and vegetable consumption. There's phytochemicals. And it seems that, again, the biophotonic scanner, even though we initially intended it to be a device to measure carotenoids, which tie in very closely with the health of your diet and lifestyle, it seems that that's now very closely connected as it's not a gene expression tester, but it's a good indication of whether or not your gene expression is probably going to be going in the right direction, right? So gene expression is very important. We can influence it, and we're applying this platform to help us have a much deeper understanding of what's better for you, a banana or a pomegranate, right? And what's it good for? So thanks, everyone. There's a glimpse.